So thank you very much, Alain, for your kind invitation. And today we talk about a story that we started years ago about the regulation of cortical plasticity, in particular during critical periods of development. During development, there are these periods that where the action of experience is particularly strong on formation of neural circuits and also on development of function. Of course, there are different critical periods depending on the different functions. Here you see for sensory systems or language. Language is maybe the most obvious critical period. You can learn a language when you're very, very young and it's very effective, the learning, but in adult it's much more difficult. The paradigm for studying this critical period plasticity was introduced more than 4 decades ago by these two guys, Hubel and Wiesel, and they got the Nobel Prize for that. And basically, the paradigm consists in a protocol of monocular deprivation that studies, therefore, critical period in the visual cortex and in the sensory system. If you do monocular deprivation for a short period of time, for instance, in a mouse, you can do a 3-day deprivation between the P25 and P28, and then you put an electrode into the primary visual cortex, you will see that the ocular dominance of the cortical neurons change, meaning that the neurons that normally were maybe preferring the stimulation from the non-deprived eye, then they become dominated by the deprived eye. So there is this shift in the distribution of the cortical neurons towards the open eye. These critical periods in the visual cortex are present more or less in all the species that have been tested so far. And you see here the critical period in cats, in rats, in monkey. Of course, there is a difference, and normally the duration of the critical period is linked to the expectation of life of these animals. So this means that there is a certain percentage of life that is dedicated to this experience-dependent refinement of cortical circuits. And interestingly, there is a critical period also for the development of visual function in humans. And this notion led to the practice of, for instance, intervening in a baby that was born with a problem of vision, with a congenital problem of vision, to intervene as soon as possible. These are the results of a meta-analysis done on babies that were born with a congenital cataract. And you see that the later the cataract was removed, the worse was the visual acuity that the eye was preserved. So if you intervene early enough, you can get some visual recovery. Otherwise, the plasticity is reduced and you don't get much of a recovery. So the questions that drove our research were why there is this reduction in plasticity at the end of the critical period. And of course, this is a cultural interest, but it's also a practical interest, because if we know this mechanism, we might devise some strategy to enhance plasticity in situations where you might want to enhance plasticity, like a recovery from a brain lesion or a neurodegenerative disease. So I'm not talking about making necessarily smarter people in normal condition, but what I mean is to have more plasticity, for instance, to facilitate rehabilitation. So, and this is when we met Perineuronal Nets. This is the staining with this lectin, WFA, Wisteria floribunda agglutinin, and this staining, as you see, is really surrounds the positive neurons. You see that the inside of the cell is black because there is no reaction, but it's really a net that surrounds neurons. And this, as you can see by this high magnification, it's really a net. A net has holes. And what is inside the holes? there are the synapses. These are the red staining is a staining for a presynaptic marker. So in these neurons, you have all these synapses that are basically surrounded by these perineuronal nets, which is therefore located in an optimal position to regulate structural synaptic plasticity. And this is just, you see, a quantification of the thing. And also, you can see this at the level of electron microscopy. If you're staying with these perineuronal nets, the cortex, for instance, the visual cortex, you will see that it's not, the perineural nets are not present on all the cells in the cortex, but they surround a specific cell type which is positive for parvalbumin. Parvalbumin is a marker of a subtype of inhibitory interneurons. And you see that the overlap is almost 100%. Maybe there are some, let's say, naked parvalbumin cells here in layer 2, but in the remaining layers, they are more or less all enwrapped by this perineuronal net. So why perineuronal nets were interesting for the studies of critical period closure? Because this staining appears in the cortex very late during development, in coincidence with the closure of critical period that, for instance, in a mouse is about P30, P40. So it's a very late developmental event. Moreover, if you do dark reading, which is a manipulation that prolongs the critical period, you also prolong this developmental maturation of perineuronal nets. Other more, causal experiment were to apply to the adult cortex chondroitinase ABC. Chondroitinase is a bacterial enzyme that cuts the sugar chains of chondroitin sulfate proteoglycans. Chondroitin sulfate proteoglycans, this complex name, I will just call them CSPGs from so far. CSPGs are the main constituent of these perineuronal nets together with other molecular entities. So by applying this enzyme, you cut these lateral sugar chains, and basically the net is dissolved and is removed from the cortical tissue. And what we got in these adult rats that were treated with chondroitinase ABC is that we could get an effect of monocular deprivation that is normally present in juvenile animals during the critical period, suggesting that the presence of this perineuronal net were closing the critical period and inhibiting plasticity. Moreover, another evidence supporting the role of perineuronal nets in this closure of critical period was another experiment in which we did actually a different, let's say, a different use of the plasticity obtained with chondroitinase ABC. Because after all, in this experiment, we actually had not made a favor to the animal. The animal has finished critical period, now we could close the eye without any consequence, and suddenly, oh no, now I have the chondroitinase and I will get amblyopia because I have the chondroitinase. But so we wanted to use plasticity in a positive way to promote recovery of function, as I said at the beginning. So what we did was to monocularly deprive rats when they were young, so to induce amblyopia, which is the visual deficits caused by monocular deprivation, and then to wait until adulthood. At that point, when you reopen the eye, you don't have recovery because critical period is closed. But if you treat with chondroitinase, we observe a recovery of visual acuity. So this plasticity that you get can be used for, let's say, good use, so for recovery from amblyopia. Of course, chondroitinase targets chondroitin sulfate proteoglycan CSPGs everywhere in the cortical tissue. And CSPGs actually form these perineuronal nets, but they are also present in a diffuse way all over, basically, the extracellular space of the brain. It has been calculated that about 2-3% of the CSPGs are really forming the perineural net. So the majority of the CSPGs are basically all around the extracellular space. So we wanted to know whether what was important for plasticity is the organization of CSPGs into a net, or just the mere presence of the CSPGs all over the extracellular space. And to try to answer this question, what we did was to study a knockout mouse where a protein which is called CRTL1 or link protein is deleted only in the brain. And this protein is necessary to keep the net stable. So in the absence of this protein, the net is unstable and has a morphological aspect that is not abnormal. This link protein is expressed again during development and with a similar temporal pattern of the maturation of perineuronal nets. So the production of this link protein probably is what is needed to organize the perineuronal net. And if you look at perineuronal nets in this knockout animal, you can see here, this is the knockout, this is a wild type, that are very disorganized, only present in this form around the cell body, but still incomplete. So with this mouse, we could ask this question. Why? Because when we measure CSPG levels, overall levels, they are normal. So we have a mouse where you have the normal level of CSPGs, but they are not organized in the net. So if the net is responsible for plasticity, we should get the enhancement of plasticity that we got with chondroitinase. If not, if there's just the presence of CSPGs, then it will be another result. And the result was that if we deprive adult knockout animals, link knockout animals, we could get an effect of monocular deprivation, suggesting that the absence of the net was what was allowing adult plasticity. So here I plotted the ratio of the amplitudes of the cortical responses recorded by means of visually evoked potentials in response to visual stimulation to each eye. And this is the ratio between the contra stimulation, the ipsi stimulation. Normally is about 2 in non-deprived animals, or in wild-type animals, deprived when they are more than P100, so they are well after the end of the critical period. And you don't see any change because critical period is finished, but you see the reduction in the contra-ipsi ratio, the contra eye was the deprived eye, in the knockout animals. Okay, so this suggests that really the net seems to be particularly important for the regulation of plasticity. Another question is that we asked was, is there an endogenous chondroitinase ABC? I mean, after all, this is a bacterial enzyme. It's something completely non-physiological. Of course, I put this in brackets because this question has not to be taken literally. There is no endogenous chondroitinase activity. But what I mean with this question is there something that could do the same job? Is this perineuronal net a dynamic structure that can be changed during plasticity, or is something that is fixed? And just by chance, by this pharmacological treatment, we are lucky enough to find the way of changing and enhancing plasticity. And so we, since there is no endogenous chondroitinase, we thought about enzymes that remodel the cellular matrix like matrix metalloproteases. But before going into this result, I want to show you this slide. This is staining for WFA, so staining for the perineuronal nets, in rats that were treated with low-dose antagonists of GABA. So these are adult animals, and by reducing a little bit inhibition, we could also enhance plasticity. And in correspondence with this treatment, we also saw a reduction in the number of perineuronal nets, suggesting that the perineuronal net is not really stable, but can be changed. Of course, this could be just the tip of the iceberg, because we are just staining the whole net, and who knows what happens at the level of the single constituents and also the molecular organization of the net. But still, this suggests us that this could be a dynamic structure, but certainly this deserves more work. Are enzymes that could do this job of remodeling the net important for plasticity. We studied, as I said, this matrix metalloproteases. And so we did this experiment. We perform monocular deprivation. Then we record from these animals, and we record separately the responses to the deprived eye and to the non-deprived eye. And we applied this broad-spectrum MMP, matrix metalloproteases inhibitor. Actually, this work was done by Maria Spolidoro, who is in the audience, so it's also a pleasure to report this data. So if you analyze the responses to the deprived eye, you see that there is the same reduction in the control and the treated rats. So no effect on the reduction of the responses to the deprived eye. So this is the period of deprivation. But surprisingly, when we look at the non-deprived eye responses that normally potentiate, so when you deprive, you have a depression of the deprived eye responses, but then eventually you have an increase in the non-deprived eye responses. You see that this increase is completely blocked. So this tells us that these metalloproteases were important for the enhancement of the responses to the non-deprived eye. And we also did some analyze dendritic spine density. We found that with this long-term deprivation that ensured this potentiation on the deprived eye, there was a small increase in the spine density in pyramidal layer 2-3 cells, and this was prevented by blocking matrix metalloproteases. So this suggests that although of course I'm not saying here that this is directly linked to the change in the perineuronal net, but suggests that changes in the extracellular matrix that can be dynamically done during the plasticity induced by monocular deprivation, and they could be important for the functional outcome of the plasticity process. Okay, so all this was done in the visual cortex, but as I said at the beginning, the final goal would be to devise some strategy to enhance plasticity in general, not only in this particular type of plasticity. So we said, why don't we try to use this chondroitinase treatment to promote functional recovery from a cortical focal stroke? So in this case, we targeted a stroke into the primary motor cortex. And by using endothelin targeted injections. And by doing this, we induce a motor impairment in the forelimb contralateral to the lesion cortex. And we did two protocols. Basically, one that we call acute protocol, because we got basically we had to train the animals to assess the motor function of the forelimb. Then we induced the lesion with the endothelin one, and in this case, for the acute treatment, simultaneously we applied the chondroitinase in the penumbra area. So this is not an overall treatment. This is to test whether cortical plasticity can be used to promote recovery in the case of these focal lesions. So we do the chondroitinase treatment to the lesion cortex, and in black here you see the reduction in the perineuronal nets that are due to the action of the chondroitinase. So the chondroitinase was really effective because it really, you see, erased all the perineuronal nets. So acute treatment, then recovery, another injection of chondroitinase, and then after 20 days from lesion, we could retest motor function in these animals. In the delayed treatment, we wanted to ask whether you really need to act immediately at the time of the lesion, or you can wait a little bit. Of course, if this is a plasticity-inducing treatment, it should also work a little bit later after the lesion, because a difference, for instance, from neuroprotective treatments, that has to be simultaneous, of course. This is the test, the motor test. It's called the skill reaching task, in which the animal has to recover this small food pellet from a very difficult position. You see, there is this very narrow window, there is this step here, so the animal has really to take the food pellet, to raise it, to put it to his mouth through the window. And so you can classify each attempt as a success or a failure, and then measure in this way the motor ability. This particular test responds to cortical lesions, to cortical lesions, because other motor function in the rat are not dependent on, they resist cortical lesions. So these are the results. These are both the raw data, meaning the positive trials, or these are the normalized, meaning that each animal is normalized to its final learning after the training period. And you see here, before lesion, the animal learn the task, so it's not something that they immediately know how to do, they have to learn it. Once they learn, we induce the lesion. And then, after the 20 days, we retest the animals. And you see that now performance is very poor in the control. So this is the effect of the stroke. What happens in the chondroitinase-treated animals? We are not preserving motor function. So if you re-examine the animals, they are just as bad as the control animals. But what we induce with the chondroitinase is the ability to have a new learning. So to use in a positive way their sort of rehabilitation, because after all, this repeated training, you could think about being a rehabilitation. So the chondroitinase promote plasticity, and probably this plasticity in the surrounding cortical areas is used to relearn this motor task. What about the delayed treatment effect? We got similar data, although you see the final learning level is a little bit less here, so it seems that the delayed treatment is a little bit less effective, but still effective. What happens in the areas that surround the lesion in terms of structural plasticity? At the moment, we only did stainings for presynaptic markers. So basically, we stain for VGLUT1, which is a marker of excitatory presynaptic terminals, VGLUT2, which label another type of excitatory presynaptic terminals, and VGAT, which stains for presynaptic inhibitory terminals. And 4 groups, we did chondroitinase-treated or control-treated, either with training or without. And basically, I can tell you that VGLUT1, no result in no group, VGAT, no result in no group. But with VGLUT2, we got an increase in the number of positive puncta, specifically in the penumbra area. Here in this graph, you have the lesion inside the lesion area, where of course you have very little terminals because everything is lesion there. And you have the penumbra where chondroitinase was injected. You have another area which is far away here in the same cortex, or we have the homotopic contralateral cortex, because in some cases you have rearrangement in the homotopic contralesional cortex. So we wanted to test whether some plasticity was occurring even there. But as I said, the only difference that we observed was in the penumbra of the animals that had received both the chondroitinase plus the training. So again, reinforcing the idea that what we do with chondroitinase is not just, allowing production of random terminals, but just opening like a window of opportunity in which environmental stimulation can promote some changes in the circuit. Of course, this is quite a, if you want, a rough way to measure plasticity, because we just measure puncta. But what we are trying to do now is to really understand which, for instance, axonal population is responsible for that, and therefore imaging axons in vivo with the 2-photon microscope. But to start to tackle this issue, we try to see where which axons were bearing this VGLUT2 positive puncta that were enhanced by chondroitinase plus training. And so we stain the axon coming from the thalamus, from the motor thalamus, and actually this population seems to be the one where these VGLUT2 positive puncta are localized. So this could be a population of axons where we could get some structural rearrangements in our case, but we will see with these different techniques what happens. Okay, so we see recovery after lesion, but what about learning and memory? Is there a role of perineural nets also in learning and memory? And as you may know, there was this very speculative paper recently in PNAS by Roger Chen that where also was thinking about a role in memory. And the first experiment studying this issue was an experiment done by Andreas Liuti and Pico Caroni. And they studied basically fear memory and extinction in particular of fear memory. Because as you know, when you induce fear memory, so you associate, for instance, a sound with an electrical shock and you create the memory for the sound, and then the animal is fearful of the sound. What happens is that this form of memory is extremely difficult to eradicate. You cannot delete it. Even if you do repeated exposure of the animal to the sound without the punishment. So the animal should learn, now it's safe. The sound doesn't predict the punishment. And for a while it works. So the animal is not afraid anymore. But then this kind of fear memory comes back. So people think that this is a model also for post-traumatic stress disorder. You see a sketch of the results by the experiments done by Gogolla, Liuti, and Caroni here. This is the fear response. When you, so after the induction of the fear memory, so you have a lot of fear, you present the sound without the punishment. This procedure is called extinction. And you see that fear goes down. But then you wait, like 1 or 4 weeks, and you check the fear response. And you see that it comes back, either spontaneously, or if you, for instance, do the so-called fear renewal, just putting the animals into the same environment in which he got the shock. So this tells us that extinction is not permanent. What they notice is that in young animals, before P23, there were papers before saying that extinction can lead to a permanent erasure of the fear trace. And again, so there was a kind of a critical period, because in these juvenile animals, the extinction was able to cancel the memory. And after a certain age, is not able anymore. And they noticed that in the amygdala, the perineural nets appear exactly at this time, when the extinction change is efficacy, is long-term efficacy. So their experiment was, we inject chondroitinase in the amygdala, and so we should restore the juvenile condition in which extinction is permanent. And as you see, this is the situation that happens in younger animals or in old animals, but treated with chondroitinase. You see that extinction, first of all, is much faster, and importantly, is permanent. So we decided to analyze, since they use chondroitinase, we decided to analyze whether, again, the perineural net was the structure that was important. And the trick to do this was again to analyze our link knockout mouse. So this is the acquisition of the fear memory trace, and you see that shock by shock, the animals get more fear, and the acquisition is the same, wild type on in the mutant. And this is the next day we do the extinction protocol. So here is the fear response after the first couple of sound, but no shock. Second couple, third, etc. First day, second day. you see that in the wild type, they remain fearful for a while, and then the next day they are still fearful, and then finally they are able to extinguish the memory. But in the knockout, you see it's very fast extinction, so very reminiscent of the effects of chondroitinase. We said, okay, maybe since this extinction was done just the day after, maybe just that memory the memory trace is just weaker in the knockout. So maybe they would have forgotten anyway. So we said, okay, if we do the extinction protocol, instead of the next day, we wait for 10 days and we repeat the extinction protocol. You see that memory is still present in the knockout. Actually, there is a trend for being even more than in the wild type. But again, we can see the fast extinction. So it's not a problem of the lack of a solid memory in the knockout. It's really enhanced sensitivity to the extinction procedure. And what about as I said, the long term? Is this extinction permanent? And it is. This is spontaneous retrieval of the fear memory, or the renewal that, as I said, means that we put the animal in the same environment to reactivate the memory of the shock. And you see that these are the wild type that are still are fearful, and these are the knockouts that, of course, are much less. We wait for 1 month and a half, and although the wild type at this point has a somewhat reduced response, but still they were different from the knockout. What could be the functional substrate of this? There are indications by the work again of many labs in this field that one possible effect of extinction is basically to inhibit the transfer of the cue information from this part of the amygdala to the more central part of the amygdala. So we did some immediate early gene staining to see activation of the amygdala, to see whether, in our case, extinction in the knockout animals was really linked to a disappearance of the activation of these nuclei in response to the cue, or there was an enhancement of this block in the transmission of the cue information. So to try to distinguish between these two possibilities, as I said, we stain for ZIF268, and we focus on the lateral and basal amygdala because even after extinction, activation should remain there in the wild-type animals. But in the knockout animals, what we observe that the density of the activated cells in the knockout after conditioning, so after the acquisition of the fear memory plus the extinction, is reduced to the level of the pseudo-conditional animals. The pseudo-conditional animals are just animals that receive both the shock and the cue, but independently. Because, of course, we had to do these control experiments because, after all, some activation by the cue itself could still activate our neurons. But we really want to know the activation that is due to the memory trace formation. And this is what you get actually in the wild type animals. So this tells us that the cue is not activating the amygdala anymore after extinction in the knockout. And the same in the basolateral amygdala. So this suggests that there are mechanisms that we are studying, we don't know the final answer to that, that allow this new learning, the extinction learning, to basically to completely erase and substitute to the fear memory trace. Okay, so another question is, that is a $1 million question and still not solved, is but how does it happen, all these things? And we said, okay, since many people has proposed that CSPGs are acting to inhibit structural plasticity, why not examining dendritic spine dynamics after treatment with chondroitinase ABC. And so what we did, we took this GFP line M mice, and we put them under the two-photon microscope. And these are adult animals. We analyzed the visual cortex, and then we treated chondroitinase to see whether there was a change in the dynamics of dendritic spines. And this was done in collaboration with Jim Mirat at the Scuola Normale, which is the twin institution of the École Normale here, but in Pisa. And so, okay, this is just the apparatus. We have this cranial window. The animal is under the microscope. You can get these images of the apical tuft of layer 5 pyramidal cells, which are the cells that express GFP in this particular line of mice. Then you can image the dendritic spines. But these are the result. So far, we have only done short-term imaging, so analyzing spine motility in spontaneous conditions. And what we observe is that by doing this time-lapse imaging, is that when you in the animal treated with chondroitinase, you have a strong enhancement of motility. So here, this is the length of a spine. And these are the oscillations, basically, in the length of the spine with time. In the chondroitinase-treated animals, basically the spine moved a lot. And many people in the field have proposed that this is the first event in the plasticity process, in the formation of new contacts. So we have an enhancement of this spine dynamics. We have an effect on the spine dynamics. We also wanted to see whether this was connected with an enhancement in synaptic plasticity. So we had this in vivo protocol of LTP induction. So we record from layer 5, we put an electron in layer 5, we stimulate the horizontal connections, and this form of LTP is very, very low in adult animals. And this is good because we wanted to have a protocol of plasticity that was reduced to see whether it could be enhanced. And actually, in the animals treated with chondroitinase, we have this enhancement of LTP. We also did another protocol which is used in slicing, which is a chemical LTP, and we applied this to the cortex, and we also got an enhancement of LTP. So there is an increase in spine motility and enhancement in synaptic plasticity. But at this point, we did another experiment. We said, why don't we do this imaging of spine motility at different times after chondroitinase application? In the previous data, these were animals that were treated 3 days before. But since when we use chondroitinase, you can really see the digestion by the imaging. So you see that it's already effective after 1 hour. What happens at this early time? Because if it's an indirect effect that is due to the digestion of the perineuronal nets around inhibitory neurons, and I want to remind you that these inhibitory neurons are aspiny, so the spines that we are imaging are not belonging to the neurons that are surrounded by the perineuronal net. So one possibility was that there was an indirect mechanism through the removal of the perineuronal net. But we thought if it's fast, maybe it's not so indirect, and it's due to the diffuse CSPG localization all over the matrix. And actually, indeed, we already see a trend for an increase in motility after 1 hour of treatment. And that becomes 3 hours later, 3 hours after treatment, you already have the effect saturated, basically, the same effect that you get 3 days. So it's quite fast. And so that's why we rethought about the interpretation of this experiment, and we said that maybe this is an effect that is due to the CSPGs and the matrix that really surround the dendritic spine. After all, as I mentioned at the beginning, these diffuse CSPGs represent the great majority of the CSPG amount in the brain. So still, I think that we should really look at the synaptic terminals onto the parvabunergic cells and to see whether structural plasticities change where the net is. This is the next, I think, experiment that should be done. From the molecular point of view, there are here, I don't think there is the need to say that the hypothesis has been that has been suggested by this work by Alain, Takao, but what was interesting is that their work suggested that the net could be a way to basically attract and concentrate some molecules, also to bridge some molecules into the parva cells, but so that the net could be a hub for different proteins. And so we, actually together with James Fawcett and Jos Verhagen in Amsterdam, they have found that there is this staining for semaphorins in the adult cortex. So when their action in axon guidance is certainly finished, there is this staining which is actually basically overlapping with the staining for the perineuronal net. And the staining increases in coincidence with the critical period closure, and also disappears in the adult when you treat with chondroitinase. So this could suggest that also these axon guidance molecules that, as I said, are present quite abundantly in the adult cortex. And so what should they there for? We don't know, basically. They could be localized to synapses, maybe to act on plasticity. That was our hypothesis. So you know very well that semaphorin 3A is a chemorepulsant, don't need to say much here. So what we did was, actually what Jos Verhagen did, was to prepare these adeno-associated viruses with receptor bodies, different receptor bodies based on the neuropilins that are receptor for the semaphorins. So we could get access to this neuropilin 1 receptor body that should bind semaphorin 3A. Since this receptor body then eventually resulted in binding also VEGF, we had to do controls because we had other receptor bodies that were binding exclusively VEGF for semaphorins. And then we used also another adeno-associated viruses that actually uses neuropilin 2, so binds to the other semaphorins. These receptor bodies were effective in blocking in vitro the action of semaphorin 3A, so they're functionally, basically they're working. So the idea is, if the net concentrates semaphorin around the parva neurons, if we counteract the action of semaphorins, we should somehow mimic what we get with chondroitinase, because we should basically cancel the effect of the net. And so the protocol was to inject these adeno-associated viruses in rats at P100, wait for 3 weeks for the expression of this stuff, and then perform the monocular deprivation and then assess the effects on plasticity. We also did some control about which serotype was more efficient, and this is the important control, staining for neuropilin 1. This is the endogenous neuropilin 1, and this is in an animal that has been transduced with one of these viruses. You see that there are, of course, not all the cells are transduced, but there is a lot of this stuff that is released, because you see that there is much more staining. And these are the results obtained with physiology. Again, here we have the contra-IPSI ratio, the visually evoked potential responses. That is normally 2, but if the contralateral eye is deprived, should go down. And we have 3 groups in which we had a significant effect, and these are the ones where we got the perturbation of semaphorins. We got effect both with neuropilin 2 and neuropilin 1, but not with the controls, and also with the receptor body that was binding only exclusively the VEGF. So I think that this experiment tells us that the perineural net is probably a complex structure that is there to collect many different proteins, but that they all go in the same direction, that is to keep the parvalbuminergic cell in a mature state, and therefore in an adult state, which also means in a less plastic state. So by this, I thank you for your attention. These are all the collaborators that have been working. Unfortunately, most of these grants have finished, and they have to be renewed somehow, but thank you.